Hey everybody, today let's go through the histology of the nephrons. The proximal convoluted tubule, as you can see here, it's like kind of associated with the venous kind of blood, and its squamous epithelium of the Bowman's capsule changes to the simple cuboidal with a lot of microvilli. The proximal convoluted tubule is the longest segment of the nephron. It fills up most of the cortex. Doesn't seem like here, but it's very coiled, And in it, there's endocytotic vesicles with lysosomes, and the apical microvilli are going to be organized in apical canaliculi. There's a lot of lysosomes because this is the first, like, the entrance of fluid, and there's of course like plenty of stuff that the body needs to excrete. So within that kind of fluid is a lot of pathogens or dust and whatever, and that's where the lysosomes are there, just to dissolve all of that. The proximal convoluted tubule is distinguished from the distal convoluted tubule because the microvilli debris is going to remain in the proximal convoluted lumen, which makes sense because you want to like excrete it out, right? So the main job of the proximal convoluted tubule is to maintain the pH of the filtrate, not the blood, the filtrate, by exchanging the hydrogen ion for bicarbonate. So other jobs that it does is it reabsorbs glucose, some amino acids, and citrate. But just recall the main dealio with the proximal convoluted tubule is to maintain the pH of the filtrate. So it secretes creatinine, like nitric bases and other bases into the filtrate, and the basal plasma membrane will form like these folds that curve in, and those contain the Na or K ATPase kind of pumps to reabsorb either water or sodium. Aquaporins are going to be the pore proteins that allow like water to come in, and most renal cell carcinomas, which is the most common kind of kidney cancer, arise from the convoluted tubule, whether it be like the distal or the proximal. So here's the renal corpuscles. This is the proximal convoluted tubule. Here's the distal. It's a little bit like larger. Here is the distal, proximal, proximal. So they're kind of like all arranged around one singular. Oh, excuse me. It's arranged around one singular vein or duct. I'm sorry, and then it's going to be lined by cuboidal cells with an apical microvilli brush border, of course, for reabsorption. And ultrafiltrate will flow from the proximal convoluted tubule down to the proximal thick descending limb, and that goes through the outer strip or stripe of the outer medulla. As it goes there, it changes into the thin descending loop of Henle within the inner strip of the outer medulla, and then from there it is lined from the simple squamous epithelium. There's no brush border anymore, so this one had a cuboidal microvilli brush border. This one doesn't. It's just back down to squamous. And then the thin limb can vary in length, and its main role is to concentrate the urine. Here is the collecting ducts. Here's the straight tubules, proximal tubules. See, the proximal straight tubules are pretty thick, and then the convoluted ones are just like all sorts of convoluted. And then right next to it is the corpuscle. Okay, so the loop of Henle is the main like ion concentrator regulator of the system, and the juxtamedullary nephrons will have longer thin limbs than of the cortical nephrons, and the ascending limb becomes the tall or the thick straight ascending limb or TAL, and that continues all the way up into the cortex as the distal convoluted tubule. The distal convoluted tubule has a initial straight part that extends a little bit to like the vesicular pole, the renal corpuscle, and then that kind of like curves around, makes the macula densa, and then becomes like the tortuous part of the distal convoluted tubule. Within the slides, both the tall ascending limb is going to be similar to the distal convoluted tubule. So in the thin descending limb, this is where water is reabsorbed. Water comes out of the filtrate. And then in the ascending limb, sodium and chlorine are going to be removed. So the distal tubule and the along with the tall ascending limb, they're going to be lined by simple cuboidal cells with no brush border. The basolateral membrane enfolding has the mitochondria in between them to actively transport, sodium or potassium in and out. Nucleus is going to be more towards like the tip, and then the thick ascending limb will communicate within the vascular end and the nephron at the outer medulla. And the distal limb of Henle is the last stage of fluid processing. So it comes from proximal, loop, and then distal. The macula densa of the juxtaglomerular complex is made out of tall columnar cells, and there's going to be an apical nuclei, basal Golgi apparatus. They have a lot of ion channels and a pretty sophisticated transporter system. And of course, these lie very close to the efferent and afferent arterioles in the mesangium in order for it to transport the ions and kind of like take away any toxins surrounding the mesangium. The cells will monitor the sodium and chloride concentration in the TAL and monitor the glomerular filtration rate. Of course, we want this to be monitored very carefully because if it's too fast, we might miss some things. If it's too slow, the body's not detoxing or excreting anything. The juxtameduloglomerular cells are going to be modified smooth muscle cells, and they line just on the outside of the afferent arterioles. And those will secrete and make renin. The juxtaglomerular cells are going to be in contact with the basal portion of the macula densa and the intima of the arterioles. Those are going to be stimulated by low blood pressure, and of course they're going to either constrict or the low pressure is going to cause the cells to constrict in order to raise the blood pressure. The collecting tubules are going to be lined with simple low cuboidal epithelium, and then those change into the taller cells into the straight cortical collecting ducts. Medullary collecting ducts are going to be lined by the columnar epithelial cells that get larger and taller as in the larger straight medullary collecting ducts of the deep part of the renal medulla and the papillary ducts. The principal light cells and the intercalated dark cells lie in the cortex and the outer medulla. So here's your proximal convoluted tubule, both in space, and it's all here's your podocytes holding everything together. And then there's the mesangial cells in the center. The principal cells are going to be responsible for water and sodium reabsorption from the lumen, and that's switched out for potassium ions. That's going to be secreted. The principal cells have the tight junction and pale cytoplasm with a few mitochondria. So the water absorption and whatnot is fairly passive. That's why there's not many mitochondria. And the apical membrane will have some microvilli with a central cilium, which is the main cilia. The main cilium to absorb is just the tallest one. It also has aquaporin 2 to allow water in, and potassium and sodium channels. The basolateral membrane have the infolding for aquaporin 3 and 4, along with the potassium, sodium, ATPase pumps, and V2 receptors. So the V2 receptors are for vasopressin. Intercalating cells are going to sort of work like the intercalated cells in the heart. They allow like coordinated conduction of electrical impulses, and it also maintains the pH balance by secreting hydrogen and absorb bicarbonate. These are going to be absent in the collecting ducts in the inner zone of the medulla, and they're going to be mostly on the outside. It blends right in because they also have like lots of mitochondria and vesicles. The type A intercalating cells are abundant. They have long apical microvilli and folds, but no cilium. This one secretes hydrogen ions and reabsorbs potassium ions. Type 2 have some apical microfolds, definitely not as deep or as it's not as deep or as evenly spaced. They're not as uniform, and they're instead of the sodium potassium pump, there's going to be a chlorine bicarb pump in order to secrete the bicarb and reabsorb hydrogen. So the descending limb of the loop of Henle allows ions and water to come in and out freely, and then the thin ascending limb is going to be only permeable to salts. The distant thick ascending limb is going to be completely impermeable to water, and then the collecting tubules involved with the concentration of urine, and they are responsive to ADH and vasopressin. So if there's no ADH, the collecting ducts are going to be impermeable to water, and that's going to dilute the urine because the body is not going to reabsorb the water. Instead, the water is just going to come on out. So the flow, just like if the water flows down. So first it goes from the urinary space to the proximal convoluted tubules, loop of Henle, ascending thin limb, then thick limb, then it goes to the distal convoluted tubules, then from there it goes into the collecting duct, and then finally to the minor calyx. So there's 3 different types of filtration. First begins with the glomerular filtration, then the tubular reabsorption, then the tubular secretion. The thickness of the layers, like from the glomerulus to the bladder, will increase. And from the minor calyx, it's going to go into the renal papillary, and the renal papillary is going to fill the minor calyx. The minor calyx is going to lead into the renal pelvis, which will lead into the ureter, then bladder, then urethra. So urine goes into from the renal papillary into the minor calyces, and each papilla drains about 10 to 25 collecting ducts of Bellini in the area corbosa. The minor calyx drains about 2 to 3 major calyx, and each will drain into the pelvis. The mucosa is transitional epithelium, and there is a urothelium, which begins as 2 layers in the calyx and then gradually increases to 8 layers in the bladder. So that's pretty interesting. And the urothelium has no submucosa. The urothelium is going to be very vascular. There is the lamina propria, made out of fibroelastic connective tissue, the muscularis propria, which is smooth muscles, and the change is that in the calyx, it's only 1 circular layer, but then it adds another layer in the ureter, and then the lower 2/3 of the ureter has even like another layer, like another outer longitudinal layer, and then of course there's about like 8 layers in the bladder. The adventitia throughout is going to be vascular, and that's going to supply blood, nutrients, and oxygens to these cells. And then the bladder and the ureter has the coverings, so they have the adventitia and the serosa. Okay, so the excretory passage occurs within like the transitional epithelium, which is about 4 to 6 layers. The cells flatten when the bladder or the ureter is distended, like blowing into a balloon. It's going to stretch out and get thinner. The apical cytoplasm has many microfilaments, and those will stay as a reserve membrane in the vesicles. And then when distended, so when the person takes an inhalation, the reserve membrane can also expand on the apical surface, allowing like more surface area. The mucosa has folds and like patches of lipids, and the muscularis propria is thicker and stronger, but poorly distinguished. Like as you can see here, you can't really tell, like, does it start here or does it start here? The middle layer of the muscularis propria is going to be the thickest, so that's the circular layer. And then there's just like a bunch of circular smooth muscle that forms the internal urethra sphincter around the neck of bladder down here, which makes sense. It needs to be thickened so it can, withstand the pressure, because it's going to bottleneck down into the urethra, of course. And in the males, it's going to have that prostate to create that bottleneck. That's it for now. Thank you for your attention, and good luck studying.